Good evening. Just confirm me that am I audible and whether the PPT is visible or not. Just make sure that everything is perfect. Let's wait for 1 minute and we'll start. Okay. Just make sure that everything is perfect. Everything is clear, right? Did you revise non-core data and core data and yesterday's topics? And for today's class, we are going to discuss about organization of genes and chromosomes. Okay. Did you revise the topic? Because today we are going to discuss MCQs related to that topic only. Okay, we'll start. Okay, and for today's class, your first question will be a mutation in LAC-I gene results in. So before that, I want to explain about an operon concept. Do you know what is operon? What is operon? See, operon is nothing but it is a group of genes, group or cluster of genes which are under the control of a particular promoter. Okay, it is simply a group of genes that are being under the control of certain promoter. Okay, that is what an operon. And my second question for you is, see, this operon will be happen in prokaryote or eukaryote or both of them. This operon takes place in prokaryote or eukaryote or both prokaryote and eukaryote. Tell me whether the operon will be takes place in a prokaryote or in eukaryote. Where this is going to happen? If you know the answer, just tell me the answer. If you don't know, just tell me I don't know. Okay. But don't be quiet. Okay. See, generally this operon will be happen in the prokaryotic organisms. Okay. Bacteria. Okay. And what is polycystronic and what is monocystronic? In case of polycystronic, one mRNA will code for multiple proteins. That is what a polycystronic. This polycystronic is seen in prokaryote. Okay, one mRNA is enough to code for a multiple protein. Okay, as it is a simpler organism, it is polycystronic. One mRNA is sufficient to code for a multiple proteins. But in case of higher organisms like human, we are monocystronic in nature. And what is monocystronic? One mRNA code for a one protein. Okay, that is what a difference between monocystronic and a polycystronic. Okay, coming back to the lac operon, it is operon will be generally happen only in the prokaryotic organisms, and there are different types of operon will be there, and we are going to study majorly a lac operon and the tryptophan operon. Okay, lac operon is said to be okay. Just tell me which operon is considered as a go operon and which operon is considered as a stop operon. Anybody know which is considered as a go operon and which is considered as a stop operon? Go operon is said to be as a lac operon or a tryptophan operon, and stop operon is referred to as lac operon or tryptophan operon. Which operon is referred to as go and stop operon? Anybody know? See, when it comes to the go operon, it is said we are calling lac operon as a go operon and tryptophan operon as a stop operon. Why? Because See, in case of a lac operon, whenever there is a substrate, here the substrate refers to a lactose. Okay, whenever the lactose is present, the operon will be turned on. Okay, the operon will be turned on inside the bacteria. Okay, that is why we are calling this lac operon as a go operon. But in case of a tryptophan operon, whenever the substrate, that is a tryptophan, is present, the operon will be turned off. That is why we are calling them as a stop operon. This is what a difference between, okay, between this lac and tryptophan operon. Okay, and you should know about the type of regulatory genes present in case of this operon concept. See, there are different three types of regulatory genes will be there: constitutive genes, inducible genes, and repressible genes. Okay, in case of constitutive genes, it is a continuously expressed genes. Okay, it will not stop. That is what a constitutive expression. For example, in our body, certain metabolic genes and ribosomes will be continuously produced. And in case of inducible genes, it is what an example for lac operon. Okay, see what I have said with respect to this lac operon. Whenever the substrate is present in the medium, the operon will be start. Okay, whenever the inducer is present, it will be induced. This is why we are calling them as the inducible genes. Okay, and repressible genes. This is what here we can say a tryptophan operon as an example. Why? Because whenever the substrate is present, whenever the tryptophan is present in the medium, the operon will be stopped. Okay, that is why it will be a repressible genes. Okay, and coming back to the lac operon, okay, see this the lac operon which have a regulatory gene, and cap site will be there, and promoter region will be there, operator will be there, and structural genes will be there. There are three different types of structural genes in case of lac operon: lac Z, lac Y, and lac A. Okay, lac Z, lac Y, and lac A. Lac Z is code for an enzyme called the beta galactosidase enzyme, and this lac Y is responsible for the permease. This is the one which permeate the lactose inside the organisms. That is what a permease enzyme. And lac A is code for an enzyme called the transacetylase enzyme. The function of this transacetylase is not well known, but we know that this transacetylase is responsible for the stability of the operon. And as the name suggests, it is going to add the acetyl group. Okay, and this is what a three different structural genes inside the lac operon. And see, we are having a promoter region, okay, and we are having the operator region, and we are having the structural genes where we can find the three types of genes: lac Z, lac Y, and lac A. And we are having the polycistronic mRNA. Okay, we know that polycistronic mRNA is seen in case of prokaryotes because one mRNA is sufficient for code for many proteins. These are all the four things important for the lac operon. Okay, and when it comes to the operator, it is the integral part of the operon. See, it is the most important concept. You should know that operon will be the integral part of the operator will be the integral part of the operon, and the repressor will be the non-integral part of the operon. Okay, see, in case of any mutation happens in the operator, the entire lac operon will be unstabilized because it is an integral part of the operon. Whenever any mutation happens in the repressor, another repressor will be replaced with this particular mutated repressor, and the function will occur normally because it is not an integral part of the operon. Okay, and this is what an important thing. And okay, now answer for this question. This is the basic idea of the lac operon. Just tell me, if any mutation occurs in the lac I gene, that is an inducer gene, it is results in constitutive expression of lac operon genes, inactivation of lac operon, enhanced repression of the lac operon, no effect on the lac operon. See, generally this I is nothing but an inducer. This is a gene for a repressor protein, where the repressor will bind here, and P is for the promoter, O is for the operator. Okay, and you know that lac Z, lac Y, lac A and all structural genes, they are code for the different enzymes. And whenever the mutation occurs in the I region, whenever the mutation occurs in this, there will be a constitutive expression of the lac operon, because the operator is never closed. Okay, see, whenever the inducer is mutated, the operator will never close. So in the if the operator is not closed, the lac operon will continuously be activated. Okay, so inducer is always going to block the operator. As this inducer is mutated, this operator will never close and the operon will be continuously turned on. So constitutive expression of lac operon genes will be happened. Okay, any doubts with respect to this question? See, before that, I want you to note the structure of this lac operon, how it looks like. See, this will be the regulatory gene. And here we can see the cap site. And here is the promoter region. And here will be the operator region. And here will be the lac Z, lac Y, and lac A. Okay. Regulatory gene is the area where we can find the RNA polymerase will bind here. In the regulatory gene, just make a note of it. In the regulatory gene, RNA polymerase will come and bind here. Okay, in the cap site, a cap molecule will be bind. Okay, what is the purpose of this cap molecule and the cyclic AMP complex? Only because when it attached to this cap and cyclic AMP, only then it will result in the efficient transcription. Okay, and the promoter where we can find the RNA polymerase will bind here, and the operator where we can find the repressor will bind. Okay, this repressor is the one going to bind with the operator. Whether the operon will be turned on or turned off will be decided by the binding of this repressor. And we know this is the structural gene. This is how the lac operon will be looking like. Now, second question: what happens to the lac operon when both lactose and glucose are present? When both lactose and glucose are present, whether the lac operon will be turned on or turned off? What happens to the lac operon when both lactose and glucose are present in the medium? See, glucose is a basic monosaccharide. We know that. And lactose will be a disaccharide, and lactose will be has to be break down into a galactose and the glucose. Only then the glucose will be taken by the bacteria. If the glucose is already present in the medium, what would the bacteria would prefer? Obviously, this glucose will be taken by the bacteria. They don't want to metabolize this lactose. So obviously, the lac operon has to be repressed. Now the repressor will be attached with the operator and the lac operon will be stopped. Okay, this is what a concept. Now, if the glucose is absent, and the lactose is absent, whether the lac operon will be turned on or turned off? Turned off. Why? Because lactose is not present. If the lactose is present and the glucose is absent, only then the lac operon will be start to occur. Okay, now it will be repressed. We know that. Which molecule binds to the lac operator to prevent the transcription? which has to be bind to the operator to prevent the transcription? Obviously, the repressor will be the one has to bind with the operator, and if it binds to the operator, the operator does not allow the RNA polymerase to move along to the structural gene. Okay, so obviously this lack of repressor will be the answer. What is the effect of tryptophan on the tryptophan operon when it is present in the environment? Here we can say this tryptophan as a co-repressor. Okay, because it is the one, if it is present in the medium, okay, definitely the tryptophan operon will be turned off. Okay, if it is present in the medium, the tryptophan operon will be turned off. That is why we are calling this tryptophan operon as a stop operon. Okay. And how this tryptophan operon will be look like? Here will be the promoter, and here will be the operator, and the leader sequence will be there, and leader sequence which will be considered as the regulatory area where we can find a different scenario. Okay. In the leader sequence, we have a different portions, and tryptophan E will be there, and tryptophan D, tryptophan C, tryptophan B, and tryptophan A. These are all the structural genes. And when 1 and 2 paired with each other, we got a passing signal. When 1 and 2 paired with each other, then the signal will be passed. Okay, if 2 and 3 will be paired, and we will get the anti-termination signal. Okay, and if 3 and 4 will be paired, we get the termination signal. Okay, when 3 and 4 will be paired, it will be the termination signal, and if 2 and 3 are paired, we'll get the anti-termination signal, and if 1 and 2 are paired, we'll get the passing signal. The signal is very important. Okay, and for this question, repression will be the answer, because this tryptophan itself is acting as a co-repressor. When it binds to the operator, definitely it is going to stop the tryptophan operon. No. Which mechanism can transposons use to cause mutation in the host genome? What is the name of the mechanism can transposons use to cause mutation in the host genome? And that will be the insertional inactivation. Can anybody know what is meant by insertional inactivation? What is insertional inactivation? See, what is transposons? What is transposons? Transposons, we are calling them as what? Very good. Jumping genes. The transposons, it is going to cause a good mutation or a bad mutation. We can say mutation with a negative impact, which has the highest priority. Always it is bad. In sometimes they are good. For example, a color of grapes, we can say like that. Mostly it will cause a negative effect. Okay, when it comes to the mutation, we can't say it is always a good mutation. Okay, now How many types of transposons will be there? How many types of transposons we can see here? There are two different types of transposons. We can say DNA transposons and we can say the RNA transposons. And this DNA transposons, we are calling them as the class 2 transposons. In RNA, we are calling them as the class 1 transposons, or we can also say that the retro transposons. Okay, and in DNA transposons and RNA transposons is there. And in eukaryotes, which transposons is very common? In eukaryotes, whether we can find the DNA or RNA transposons. Very good. In eukaryotes, we are going to find majorly generally we can find both the type of transposons, but RNA is very common. But we can say both DNA and RNA transposons will be there. But class 1, that is RNA retro transposons will be very common in case of eukaryotes. But in prokaryotes, only class 2, that is DNA transposons is possible when it comes to the prokaryotes. And in we are saying this class 2 transposons and class 1 transposons, and in class 2 we have two different types of DNA will be there. Okay, replicative DNA and non-replicative DNA. Okay, replicative DNA and non-replicative DNA. Replicative DNA, and which is called as the copy and paste transposition, which is called as the copy and paste transposition. Replicative will be the copy and paste transposition. Very good. And in which DNA, whether it is a replicative DNA or non-replicative DNA, the total number of copies will increases. In which DNA, the total number of copies will increase. In replicative DNA, it is called as the copy and paste transposition. Here, the total number of copies will be increases. In non-replicative DNA, it is a cut and paste transposition. Here, no copies will be increased. Okay. Now, see, transposons can disrupt the gene function. Okay. See, we are calling them as the jumping genes. Simply, some of the genes will jump into another area, and it is attached with and it is mingled with the other gene where it is not needed. Okay, and it is inserted and it is coding for a protein, and that is called as the insertional inactivation. Okay, this insertional inactivation, this insertion of some genes where it is not needed, that particular gene leads to a mutation. Okay, this is what an insertional inactivation. And which level of chromatin organization involves the wrapping of DNA around the histone octamers? Come on. Just tell me which level of chromatin organization involves a wrapping of DNA around the histone octamers. And what is the function of this histone protein? Acetylation, it is the one going to wrap up the DNA. Okay, when it is attached, when it attached with the DNA, it makes the DNA into a condensed form. Okay, what we are calling them as the chromatin. Okay, and what is the charge of DNA? What is the charge of DNA? Positively charged or negatively charged? Negatively charged. Why it is negatively charged? Why it is negative charged? What is present to make the DNA to be negative? Why it is negative? Very good. Because of the presence of phosphate group. And what is the charge of histone protein? What is the charge of histone protein? Positive charge. Why it is positive? Which amino acid make the histone protein positive? Very good. Because of the presence of lysine and arginine. Okay, it makes the histone protein positive. So both the charges are getting attracted to each other, and histone protein makes the DNA a most condensed form. Okay. Now, and also histone is the one going to wrap the DNA, and they are the one converting the DNA into euchromatin as well as the heterochromatin. And heterochromatin is the most condensed form. Euchromatin, in the heterochromatin we can't find the transcriptional site, so the promoter will not bind to the initiation region, so the transcription will not going to happen and the protein is not going to synthesis in case of heterochromatin. But in case of euchromatin, the transcription it is not in a condensed form. Instead of that, it is in a relaxed form, and where we can find the transcriptional site. Okay, it is free, so that the promoter will bind in the initiation region and the protein synthesis will be continued. Right? And now answer for this question. See, always which level of chromatin organization involves the wrapping of DNA around the histone proteins? That will be the secondary structure. Okay. And what is nucleosomes? See, this secondary structure of chromatin involves the wrapping of DNA around the histone octamer. Always the histone will be occur in the octameric form. Kindly make a note of it. This is an important statement. And when it is wrapping the DNA, it is converting them into the nucleosomes. Nucleosome is considered as the lowest level of chromatin organization. Okay, it is a lowest level of chromatin organization. It is the one pack the DNA in the stable coiled form. Okay. Which histone modification is associated with transcriptionally active chromatin? And before that, you should say what is known as the active chromatin. What is active chromatin? What will be the technical term? Active chromatin. Very good. And what will be the modification will be happened in case of active chromatin? Quick. Why are getting so much of time for this question? Acetylation. Okay, for the acetylation is the name of the modification, post-translational modification. It is associated with the transcriptionally active chromatin, that is euchromatin. See, this acetylation of histone tails, this is the one acetylation. It is neutralizes their positive charge. Okay, by neutralizing these positive charge, they are going to loosen the chromatin structure. So what makes the heterochromatin to convert into the euchromatin means this modification. What is that? Acetylation. Okay. Now then only the RNA polymerase, that is a promoter, will associated with the initiator region. Then the transcription will be continue. mRNA will be synthesized. Okay. Now, what is the function of the heterochromatin? We know that heterochromatin will be the most condensed form and euchromatin will be the loosened form. And what is their function? Why it has to be exhibited in a condensed form and the loosened form? You can easily eliminate the option, right? It is condensed and transcriptionally inactive. Okay, it is characterized by tightly packed nucleosomes. It makes the DNA in a stable form to maintain the chromosome structure and their stability. Okay, what is the primary role of heterochromatin in the nucleus? In the nucleus, what is their primary function? Facilitating transcription, DNA replication, chromosome segregation, repairing DNA damage. Quick. Facilitating transcription. See, just now we have said that it is a transcriptionally inactive. Okay, it is transcriptionally inactive. Its primary function is, as I said before, it has to maintain the structure and stability, and also it is involved in the chromosome segregation. Okay, chromosome segregation. It has no role in the DNA replication, no role in the transcription, no role in the repairing DNA damage. Okay. Which histone proteins? Kindly read the explanation also. Okay, it is this heterochromatin plays a crucial role in the cell division process. Okay, by helping to organize and compact chromosomes, ensuring their proper alignment and distribution to their daughter cells. Because one cell has to be divided into two cells in the mitotic division, is it not? And the complete proper segregation between these two daughter cells will be done only by this heterochromatin. Okay, so home chromosome segregation will play an important role. Which histone modification is commonly associated with the heterochromatin formation, or we can say the transcriptionally inactive chromatin? Which histone modification is required for heterochromatin formation? Okay, very good. Methylation will be the histone modification associated with the heterochromatin formation. Okay, see, methylation happens in the histone tails. This is the one leads to the chromatin has to be condensed and the transcription will be repressed where it is inactive. Now, which of the following disease is associated with the heterochromatin abnormalities? Which disease is associated with the heterochromatin abnormalities? Maintaining a structure of chromosome and its stability is very important, and that has to be done by the heterochromatin. If the heterochromatin is not functioned normally, what will be the disease will be happen? Is it so? And that will be the Rett syndrome. Okay, it will be the neurodevelopmental disorder. This is the one caused by the mutation, and this mutation occurs in the MECP2 gene. Okay, this is the gene involved in the chromatin remodeling and the heterochromatin formation. If the mutation happens in this gene, means it results in the Rett syndrome, which results in the neurodevelopmental disorder. Okay. Now, what distinguishes constitutive heterochromatin from the facultative heterochromatin? Constitutive heterochromatin and the facultative heterochromatin. First option, constitutive heterochromatin is always transcriptionally inactive, while the facultative heterochromatin can become active under certain conditions. And constitutive heterochromatin is found near the centromeres and telomeres, while the facultative heterochromatin is dispersed throughout the genome. And third option is constitutive heterochromatin contains the repetitive DNA sequences, while the facultative heterochromatin contain unique gene sequences. Option D, constitutive heterochromatin is replicated during the S phase and facultative heterochromatin is replicated during the M phase. What will be the answer? Okay, see, very good. Constitutive heterochromatin is always transcriptionally inactive. Constitutive means continuous, okay, always. And heterochromatin means condensed form. Always it will be in a condensed form, which is transcriptionally inactive in always. And while facultative heterochromatin will be active under certain conditions. Okay, and this constitutive heterochromatin is permanently condensed always, and the facultative chromatin can undergo a reversible changes in their structure. Sometimes it will be transcriptionally active and at many times it will be inactive. It is reversible, okay, and it constitutive will be a permanent. Which of the following is not a characteristics of euchromatin? Why some people are not answering? Everybody should answer even though it is right or not. Okay. See, it contains mostly a repetitive DNA sequences will be the wrong statement when it comes to the euchromatin. We know that euchromatin will be in the loosened form and where we can find the transcriptionally active sites will be there and promoter will bind to the initiation region and the mRNA synthesis will be continued. And it is loosely packed, we know that, and it is enriched in the gene and regulatory elements. Only that we can call them as the transcriptionally active region. And it contains a unique protein coding sequence rather than a repetitive DNA sequence. This is the most important difference between the heterochromatin and euchromatin. Okay. Now, what is the primary role of euchromatin in the nucleus? For heterochromatin, the primary role will be their chromosome segregation inside the nucleus. And in euchromatin, what will be the primary role in the nucleus? Facilitating chromosome segregation, DNA repair, transcriptional regulation, chromatin condensation. Okay, and transcriptional regulation will be the correct answer. I hope I don't want to explain much. And what is the difference between and what distinguishes this facultative heterochromatin from the constitutive heterochromatin? Again, the same question like the heterochromatin. Facultative euchromatin is permanently condensed, while constitutive euchromatin can undergo a reversible changes in their structure and transcriptional activity. Facultative euchromatin is enriched in the repetitive DNA sequence, while the constitutive euchromatin contains mostly protein coding regions. Facultative euchromatin is found near the centromere and telomere, while the constitutive euchromatin is dispersed throughout the genome. Facultative euchromatin is replicated during the M phase of the cell cycle, while the constitutive euchromatin will be replicated during the S phase. Okay, see, the facultative euchromatin is permanently condensed. Okay, while the constitutive euchromatin can undergo a reversible changes in their structure and the transcriptional activity. This is what a difference between the facultative and constitutive euchromatin. So the facultative euchromatin will be transcriptionally active or inactive. Okay, based on their need, it will be transcriptionally active or inactive, whereas the constitutive euchromatin will be continuously active. Okay. Which of the following disease is associated with the abnormalities in euchromatin structure or function? Fragile X syndrome, Angelman syndrome, Prader-Willi syndrome, Williams syndrome. Hello, answer for this question. Which disease is associated with the abnormalities in euchromatin structure or function? And that will be the Fragile X syndrome. Okay, Fragile X syndrome will be the correct answer when it has the mutation in FMR1 gene. Okay, which leads to the abnormal methylation and heterochromatin formation in the promoter region of the gene. This results in the transcriptional silencing, and mainly the mutation in this gene results in the Fragile X chromosome. And what percentage of human genome is estimated to consist of unique DNA? It will be less than 1%. Okay, while the human genome is largely composed of the repeated DNA element. Okay, and unique DNA encompasses the protein coding gene, regulatory genes, and other non-repetitive genes, or everything will be coming under unique DNA, and that will be comprises less than 1%. And what is the primary function of unique DNA sequence in the genome? You have furthermore 7 questions. Be quick. And before that, I have to explain what are all the types of DNA sequence we can find in our body. See, DNA sequences can be categorized into 2 types. One is unique DNA and unique DNA, or we can say the non-repetitive DNA and the repetitive DNA. In the unique DNA, we can categorize further to truly unique. Okay, which will be a single copy of a gene, and this will be the gene family where we can find the similar sequences, and hemoglobin will be the example. And in hemoglobin, we can call them as the globin gene family. Under that, we have a cluster of related genes like hemoglobin. And in hemoglobin, we have two alpha and two beta. And in case of embryo, we can find two zeta and two epsilon. These are all the types of unique DNA, particularly the gene family. Similar sequences can be seen. And in repetitive DNA, we have two different types of repetitive DNA. One is a moderately repetitive and the highly repetitive DNA. In the moderately repetitive DNA, we can further categorize into two. One is tandem repeat and another one is interspersed repeat. In the interspersed repeat, we can find the sign and the line. Okay, SINE and LINE, sign and line. In highly repetitive DNA, we can find and satellite DNA and mini satellite DNA and microsatellite DNA. And what is your duty is how many base pairs can be seen in all these type of DNA is very, very important. See, in case of moderately repetitive DNA, we can find approximately 150 to 300 genes. 300 base pairs will be seen in case of moderately repetitive DNA. And in case of highly repetitive DNA, we can find less than 10 base pairs. Less than 10 base pairs of the DNA base pairs will be seen in case of highly repetitive DNA. And this highly repetitive will make up the 11% of the human genome. Okay, are you clear? Okay. And what is the answer for this question? See, this flow chart is very important. Kindly make a note of it. And the primary function of this unique DNA will be the regulation of gene expression. Okay. Which of the following disorders is caused by the mutation in unique DNA sequences? If in case any mutation happens in the unique sequence of DNA, unique sequence DNA, what will be the disorder will be resulted? And the disease will be the Duchenne muscular dystrophy. And what is meant by Prader-Willi syndrome? Do anybody remember what is meant by Prader-Willi syndrome? What is it? And which chromosomal aberration which causes this Prader-Willi syndrome? Which chromosomal aberration? Whether it is a deletion or duplication or inversion or translocation, which chromosomal aberration which causes the Prader-Willi syndrome? We have a 46 chromosome, and in 15th chromosome, particularly the long arm at the 13th region, where we can find the deletion. This condition results in the Prader-Willi syndrome. Clear? Kindly make a note of it. We have a 46 chromosome, and what is the type of chromosomal aberration? Deletion in the 15th chromosome in the region of the short long arm on the 13th region. Okay, this is why the condition results in the Prader-Willi syndrome. And when it comes to the mutation in unique DNA sequence, it results in the muscular dystrophy. And which of the following is not a type of repetitive DNA sequence? Just now I have explained the type of DNA sequence. If you listen carefully, you can easily answer this question. Not a type of repetitive DNA. Okay. What about the others? Protein coding genes are not considered as the repetitive DNA sequence. Okay, see this repetitive DNA sequence includes a satellite DNA, ALU elements, telomeric repeats, and other repetitive genes. Okay, which repetitive DNA sequence is commonly found in the centromeric region of the chromosome. It is found in the centromeric region of the chromosome. Centromeric region of the chromosome. Which repetitive sequence will be seen in the centromeric region? Okay, that will be the satellite DNA. It is the one found in the centromeric, particularly the heterochromatic regions of the chromosomes. Again, it also involved in the chromosome structure and their stability. And which repetitive DNA sequence is involved in protecting chromosome ends from degradation and fusion? The most important question. Okay, telomeric repeats, because it is the one maintaining the length of the DNA. Okay, by the DNA, it is frequently undergoes a degradation process. There comes the telomerase enzyme, which is adding the sequence called this at the end of the chromosomes to maintain its chromosome stability. Right? And which of the following technique is commonly used to visualize the organization of genes and chromosomes? Which technique is commonly used to visualize the organization of genes and chromosomes? Quick, quick, quick. Fluorescence in situ hybridization. Okay, FISH. It is the one, a technique, a cytogenic technique to visualize the organization and location of specific DNA sequence on our chromosomes. Okay, so read about this technique. It is very important. Their working phenomenon is important. And which of the following accurately describes, describes the organization of genes and chromosome in the human genome? Accurately describes the organization of genes and chromosome in human genome. Each chromosome contains one gene. Genes are organized linearly along the chromosome with non-coding regions interspersed. All genes are clustered together in the genome. Chromosomes are composed entirely of a repetitive DNA sequence. Oh, each gene contains only one, each chromosome contains only one gene. Genes are organized linearly along the chromosome with non-coding regions interspersed. This is what a perfect definition for this organization of genes and chromosome in the human genome. Finally, which of the following, okay, the same question. Okay, fine. That's all for today. For tomorrow, what is the topic? Cell division. Cell division is the topic you have to read for tomorrow and tomorrow's test. Everybody clear? Do you have any doubts? You can wait or else you can leave the class. And for tomorrow, you have to read and count the cell division, cell cycle and cell division. Okay, fine. Thank you. And if you have any clarification, you can post it in the group at any time. Okay, fine. Thank you.